Good evening, everyone. It's so wonderful to be back in this room with all of you for the first Crossroads of Ideas for the 2021-22 academic year. My name is Laura Heisler, and I'm the Director of Programming for the Wisconsin Alumni Research Foundation, the Director of Outreach for the Morgridge Institute for Research, and a partner with the Wisconsin Institute for Discovery. And these three organizations collaborate to bring you this series, which is all about issues that are the subject of research at UW-Madison and also that happen to be really important to all of us in our lives. Before I get on to talk to you about some upcoming events you might be interested in and tell you how this evening's going to go, I want to take a moment and hope you'll join me in acknowledging the land that we find ourselves on, those of us here in the room, and possibly some of you at home. The University of Wisconsin-Madison occupies ancestral Ho-Chunk land, a place their nation has called Teejop since time immemorial. In an 1832 treaty, the Ho-Chunk were forced to cede this territory. Decades of ethnic cleansing followed when both the federal and state governments repeatedly, but unsuccessfully, sought to forcibly remove the Ho-Chunk from Wisconsin. This history of colonization informs our shared future of collaboration and innovation. Today, UW-Madison respects the inherent sovereignty of the Ho-Chunk Nation, along with the 11 other First Nations of Wisconsin. Before we get on to how this evening's going to go, we have a ton of events coming up that I think you might like to know about. So in chronological order of the next events, next week in this room and on Zoom, we will be featuring the first night of the Wisconsin Science Festival. And we have a very popular event that has become a hallmark of the festival called Big Ideas for Busy People. This year's topic is all about how do we know what we know, the role of evidence in advancing understanding. Eric Wilcots, who'll be your moderator tonight, is also moderating that session. Big Ideas for Busy People is 5-minute power talks. And they won't be longer than 5 minutes because we have a giant gong. We gong them right off the stage if they go longer. So I hope you'll join us for lots of different perspectives on this question from UW-Madison researchers. Also, as part of the Wisconsin Science Festival next week, Larry Shapiro and Steve Nadler, who brought this series, it's a session that we have tonight to you, are going to be part of the Wisconsin Book Festival, Wisconsin Science Festival collaboration, again in this room, talking about the recent book, When Bad Thinking Happens to Good People. And then again, the Crossroads of Ideas series will continue. Next month, we have 2 sessions, actually. We are doing a special session in partnership with UW Communications and the Science Writer in Residence on COVID-19 and how to interpret all of the data that have been coming from that, from the study of the pandemic. And then the Big Questions series that we'll be talking about in a moment continues the week after with another one of the big questions, what is consciousness? And to say more about how this whole series is going, I'm going to be introducing Larry Shapiro in just a moment, but a few housekeeping notes for you. Crossroads always features a Q&A, and we'll be doing that tonight. And those of you watching at home, you'd be welcome to ask questions as well. Just use the Q&A feature on Zoom, and I will be monitoring those, and I will let Eric, our moderator, know when we have a question online that we want to ask, and I'll just read it out loud from the microphone. Those of you in the room, we have a very COVID-safe mechanism. We ask that you line up in front of the mic here rather than pass the mics around. So that will be at the end of the session. My suspicion is we may not end exactly at 8. So if you need to leave a little early, please do. I'm also guessing the speakers will stick around a little bit after if you have questions and you didn't get a chance to ask them. So with that, I'm going to turn things over to Larry Shapiro, the Baron Ensch Professor of Philosophy here at UW-Madison, to tell you more about the Big Questions series and tonight's session.
Well, I guess I should first address the elephant in the room. I know I need a haircut. Thanks, Laura. The idea behind this series of talks was that it'd be fun to pair a philosopher with a scientist. People not hearing? It'd be fun to pair a philosopher with a scientist to talk about an issue of fundamental importance to the human experience. And so tonight we'll be listening to a philosopher, Carol Cleland, and a scientist, John Yin, talk about what life is. And our next conversation will be about consciousness. And then the spring semester, we'll tackle the big questions about whether we have free will, whether there are moral truths, and what is happiness and why, or is it important? Before we get started tonight, I just wanted to offer thanks to the many people who are making this series of talks possible. We have Laura Heisler, who's the Director of Programming from WARF. We have Steve Nadler, Director of the Institute for Research in Humanities, and his Associate Director, Katie Apsey. Joe Handelsman, who is the Director of WID, has offered us this nice space and helped us frame these conversations. And Becky Balistreri, who's the administrative assistant for WARF. All of these people have worked very hard to make this evening possible, as well as future evenings. And I'd like now to introduce Dean Eric Wilcots. Dean Wilcots is the Dean of the College of Letters and Science, but he's also the Mary C. Jacoby Professor in the Department of Astronomy.
Good evening, everyone. Thank you, Larry, for the introduction. It's great to be here. Great to see you all, and it's my honor to introduce our two speakers this evening. So, Dr. Carol Cleland is a full professor of philosophy at the University of Colorado-Boulder. She received her BA in mathematics from UC Santa Barbara and her PhD in philosophy from Brown University. She's unusual among philosophers for both the breadth and the depth of her scientific knowledge, including the fields of microbiology, astrobiology, and the origins of life. She's been affiliated with the NASA Astrobiology Institute and is currently an affiliate of the SETI Institute, SETI standing for the Search for Extraterrestrial Intelligence. At UC Boulder, she is a co-investigator in the Center for Astrobiology and the director of the Center for the Study of Origins. She is the author of The Quest for Universal Theory of Life, Searching for Life as We Don't Know It, published by Cambridge University Press in 2019. She's also an avid skier, although she told me not to tell you that. And we have similar roots in the great state of Pennsylvania. So it's great to have Carol join us this evening. Our other speaker tonight is Dr. Jonathan Yin. Dr. Jonathan Yin is a Vilas Distinguished Professor of Chemical and Biological Engineering here at UW-Madison, as well as a faculty member at the Wisconsin Institute of Discovery, so the building that we are in. He holds a BA and BS from Columbia University and his PhD in chemical engineering from UC Berkeley. Also is a cellist of some note. His primary research focuses concerns on the growth of viruses, where this requires understanding interactions between the virus's genome and the environment that the cell hosted. Study of viruses has led Dr. Yin to consider which chemistries are necessary for the origin of life, discovering that the presence of certain chemicals would facilitate the merging of amino acids, those building blocks of life, in conditions that would have been present during the Earth's early history. This discovery was published recently in the journal Origins of Life and Evolution of Biospheres. And so with that, I think, Carol, you get to go. Who's going first? Jonathan's going first. So please welcome Dr. Jonathan Yin and Dr. Carol Cleland.
Thanks very much for that kind introduction. I'd like to thank the organizers of the Crossroads activity and Laura Heisler for organizing, as well as Larry Shapiro, sorry. So what is life? If we look at these various images, I think most of us would not have too much trouble discerning those that are living from those that are not. But you're not interested in those sorts of questions. We're trying to look for something a little bit deeper. So when we look at life, we see a huge amount of diversity, and the hope is to try to get at definitions that encompass this diversity. Normally, I like to think about big picture, holistic perspectives on science, but I think this is an exceptional case where the reductionist perspective is helpful. So if we want to think about life from our, if we look in Wikipedia or dictionary.com, we've got a definition of life as distinguishing phenomena of organisms, especially metabolism, growth, reproduction, and adaptation to the environment. That's fine for those plants and animals. But if we look to reductionist approaches, we might start looking at cells, molecules, and the atoms. So underneath the words here, you see cells, molecules, and atoms. On this figure that shows over many orders of magnitude, some might know the powers of 10, where we've got a human here and much smaller, zooming in on the hand and the cells in the hand, the tissue and the cells and the molecules. I guess I should be pointing with this, molecules and ultimately atoms. So to get at the ideas of life, it's useful to think about cells, molecules, and atoms, because this is where a lot of life converges. And that's where we have lots of commonalities. So here's a definition of life from NASA. John Rummel was looking to get some advice from experts in the field back in 1992 in this early exobiology program, seeking to understand life perhaps elsewhere. And this panel came up with a definition of life as some self-sustained chemical system capable of undergoing Darwinian evolution. So it homes in on chemicals. These are then at the scale of molecules or atoms that are able to somehow self-sustain. So you think about chemical reactions that are able to keep on going, and also are able to somehow evolve. So we have a concepts of how animals and plants evolve, but Darwinian evolution in a chemical sense is a harder concept to grasp. So maybe it's helpful to look at a little bit of history here. Here we are, present day, and here is Luca, the last universal common ancestor from some 3.6 billion years ago. All right, this is the last universal common ancestor implies this is what we share with all other life on this planet, that we all are descended from this particular life. And the way we know this is by looking at the particular genes that we all share with all life. This is some 300 genes. I think Carol will go into this in greater detail. I'm going to actually not discuss Luca, but go back a half billion years further to this stage of prebiotic chemistry. I should have gone all the way back here, that 4.5 billion years ago, we had the formation of the Earth. So we're moving into this realm of 4.2 to 4 billion years ago before the present. And this is the realm of prebiotic chemistry, where we've got molecules hopefully doing interesting things. And that's something that we're trying to coax in our lab, molecules to do interesting things. So I'll try to frame tonight's question from one aspect of our work as a chicken or egg problem of prebiotic chemistry. We're all familiar with the concept of genetics. What's the information that our parents gave to us? And that information is played out as we develop. It affects our hair color or what sort of metabolism we might have. But there's also the metabolism, which is, well, you probably had some meals today, and because of those, you're able to hopefully stay awake during these talks, and that sort of energy processing is important for you. So before I go into the details, I should mention the people who actually do the work. I'll primarily be talking about work from Isabella Sibilska-Kamensky. And she was a postdoc in the lab several years ago. Currently, the work is going on with Haley Boyganjan, who is a chemical engineering PhD student. But we have a lot of help from colleagues around campus, particularly in the analytical chemistry from Professor Lingjun Lee in pharmacy and chemistry, and her students. People who are interested in chemical and microbial ecology, Professor David Baum and his student Lena Vincent, Professor Venturelli in biochemistry. We're also interested in network inference and dynamics. How can we better understand how molecules are interacting? And here, people who are in bioinformatics and computational biology are helpful. This is also a colleague of my own, in chemical engineering. Victor Zavala and his co-workers are helping us with some of the modeling. So we do both modeling and experiments, and we're trying to, modeling meaning building computational models of what could be happening to help interpret and guide our experiments. And we should, of course, thank the funding. Underneath here is also Wisconsin Alumni Research Foundation, who are sponsoring tonight's program. Okay, so let's move on. Which came first, genetics or metabolism? Or more generally, we can think of which came first, and information or energy, information processing or energy processing. We do both as living entities. So I'll also point out that as residents of Wisconsin, and in particular Madison, you're aware already that phosphates are important for life, because we're cautioned to keep the leaves out of the streets, because the phosphates will make this very rich tea that gets into our lakes and causes algal blooms. So we're aware that phosphates are great for life, but including life that we're not so interested in promoting. One kind of phosphate that one sees a lot in biology is called ATP. The P stands for phosphate, and there are three of these. The T stands for triphosphate, and the A is this other stuff. So this will be a player in, this is a kind of currency that we see a lot in biology. So I need to go into a little bit of the biochemistry. I promise I won't make it, I won't have a quiz, and I'll try to make it as painless as possible. Key aspect here is that when you take a bite out of that dinner or breakfast or donut, then you are feeding glucose or sugars into your system. And your system, your cells that metabolize this glucose go through a variety of different processes, many arrows here, but key here are the arrows that produce ATP, shown in the yellow blocks. So when we break down the glucose, we are combusting the glucose, there's oxygen involved, and we produce as byproducts carbon dioxide, water, and this ATP, the energy. So you're able to pay attention now because you have ATP being generated, and that's keeping you awake, hopefully. Okay, so these metabolic reactions are making ATP by combustion of glucose. Now, if we home in on a little bit of this figure, just this one arrow right here, there's a lot of stuff going on in there. So I want to home in on that particular arrow. And here it is. That lot of stuff are these various other steps, many other arrows within there. So now I home in on the very first arrow. When you take that bite out of your donut, that glucose then experiences hexokinase. So hexokinase is a large molecule, this big blob here, relative to the glucose molecule. The glucose gets into the claw or the cleft of this enzyme, and it gets a reaction done to it. It gets phosphorylated. I'm sorry, something strange happened there. It gets phosphorylated, or a phosphorus group gets plugged in there. And and this consumes ATP. Later on, we make ATP. But this is a first step. And the key point here is that in order to make ATP, we have to use these various large proteins or enzymes that are able to break down and start doing reactions on the glucose. So these metabolic reactions are catalyzed by these proteins. They are facilitated by that. Okay, so where's the chicken or egg problem? Well, I've just shown you how the protein helps us to make ATP. I haven't shown you the other part, we're limited on time, but likewise, when you make this big molecule here, you have to feed in ATP. The molecule here is a string, beads on a string that for each bead, as it gets connected to the next one, these are amino acids, ATP gets invested. So that's the chicken or egg problem. You need to make ATP, and that's what this large molecule facilitates, but ATP needs to be invested in order to make that molecule. How could such a process of information and energy processing ever get started? This is the energy processing making ATP. Here's the information processing that ATP helps to get the information out of the DNA in your cells into making this particular enzyme. So this is about the closest Darwin came to telling us about what he thought of the origins of life. And this was in a letter to Joseph Hooker. But if, and oh, what a big if, we could conceive a small, warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc., present, that a protein compound, think about that big enzyme, was chemically formed, ready to undergo still more complex changes, all right? So that was Darwin's intuition that maybe you could take relatively simple chemical compounds, provide some energy, and come out with some sort of complex machinery that would give us some insight into perhaps how life would come about. Well, there's a famous experiment that was done in 1953 down the street at University of Colorado. Stanley Miller, as a graduate student, said, let's try putting together some compounds, simple gases here, ammonia, methane, hydrogen, no oxygen. And that's our primitive atmosphere. We're going to have an ocean here, some water. We're going to boil that up, and we're going to add some energy source, spark. And so he did this, and underneath here, there are amino acids that were found as products. So amino acids are the building blocks of the enzyme. So those beads on a string, the amino acids are the beads. And this is in many high school textbooks on origins of life, the Miller-Urey experiment that showed how you could cook up simple gases and get the building blocks of these proteins. So I've always, I think this is a wonderful experiment that suggests at least an early step in prebiotic chemistry. Unfortunately, when you look at the next page in these high school textbooks, they don't tell you what happens to the amino acids. And presumably then cells and other things came about, but those details are missing. So that's largely where the rest of prebiotic chemistry has been the last 70 years, trying to figure out how to go from these simple molecules to more complex ones. So I show here two amino acids, the various simplest ones. This is a little bit of chemistry, sorry about that. But what we'd like to think, you can think of them as different colored beads, which we'll name one G and one A, one for glycine and alanine. There's amino, think about nitrogen or ammonia, there's a nitrogen in there, or acid, carboxylic acid, think about vinegar. When these two get together, you can get a reaction that allows condensation to occur, which means water comes out and you form a bond between these two beads. And we call that GA, or the chemical term is glycosylalanine. So this is a chemistry that allows these two beads to be bound together with a very strong bond. And the protein that I showed, the very complex protein molecule, might have several hundreds of these beads connected together. It folds up and then it has various activities. So this is all well and good, except principles of chemistry tell us that when we've got an excess of water, as we might have in a small little warm pond, then the chemistry does not promote beads coming together, but beads coming apart. So if you put the protein in water, over time, it'll break up. This is a challenge for Darwin, because in that warm little pond, we would not tend to we would not observe beads coming together. So we thought that maybe one approach would be to remove the water. Let's try to put the amino acids in water, but allow them to dry. So we dissolve amino acids in water. This is in a test tube. We set the pH, this is the acidity, and temperature. We incubate for 24 hours, open to the atmosphere, and we get a solid in here. Then we rehydrate and we analyze. It turns out that this solid is quite reactive. It can allow these strings to come together. So in that view, it'd be nice if we can understand how the strings are coming together. And that's where we go to our analytical chemistry friends who have ways of helping us to guiding our experiments to actually analyze for something like, here's G and A, what we started with, but there are things like G2 or AA or AGG. So various strings coming together, 2 or 3 or 4, we have ways of detecting them. So we knew from the literature that people could set temperature and pH and have different effects, and we didn't know what temperature or pH to set. So we thought, let's try to examine a whole lot of different temperatures and pH. So most of you are sitting in the room with a pH of 6 or 7 in your blood, and your temperature is about 37 degrees Celsius. So you're right about here, your blood. But if you go to look at your stomach acid, then your pH is more like pH 2 or 3. Or if you go and get some Drano, then it's more like pH 11 or 12. And you know, if you boil some water, then you're up here 100 degrees. So here we have for 11 by 12, 132 different environments where we decided it might be interesting to run the reaction under those different environments. So again, we also thought, well, you know, this energy part, ATP is oftentimes a player, and ATP is known to be able to help facilitate these bonds coming together. The A part, though, of ATP is hard to imagine how this could be coming together very easily from, it's much more complex than the amino acids. So we use something that volcanoes can create, a different version of triphosphate. This is a cyclic version that doesn't have all the organic chemistry on the end. It turns out this TP or trimetaphosphate can also work just as well. So we wanted to study different temperatures and pH in the presence and absence of trimetaphosphate. In the absence, no TP present. Here's the production of diglycine, stringing together the very two simplest amino acids. And what you find are lots of white regions where we did not detect any products. So everything below 30 degrees C, that's room temperature and below, we didn't see any reactions. Well, at that level, we don't evaporate the water. Water's still around. There's no activating agent, no chemistry. We have to go to high temperatures like boiling points or above 70 degrees C. And at high pHs, think about that Drano, or low pHs, think about that stomach acid. These are not very hospitable conditions, all right? But those are the conditions where we saw these two balls coming together. What if we have TP present? Well, when we have TP present, then we get this kind of a profile. This is, again, diglycine forming, the two beads coming together, but we have beads coming together even at temperatures in which we don't evaporate. So water is present, and the activating agent can enable those balls to come together, and we get a lot more reactions and products. This is just a difference between the two plots, with the black being the minus TP and the blue being plus TP. So we're seeing a lot more products when TP is around. We looked at many more products than just diglycine. We looked at, we're able to look at quite a few. And so here's alanine glycine, two different balls coming together, similar sorts of patterns. If we go to 3, then we see the 3 making the 3 ball or 3 amino acid peptide. We also make small amounts in the absence of the activating agent and much more in the presence of the activating agent. I like this experiment in particular because these two molecules, AGG and GGA, have the same chemical composition, just a different sequence. Think about 100 and 001. Those are information strings. And what's the information that's encoded there? Well, these formed under these particular environments. So in a particular environment with these particular 1s and 0s, you get different strings forming in the sense that the information is encoding the environment, and we can see it in the products that are formed. Okay, So I think the take-home message is here, in the absence of TP, we've got molecular strings that arise under harsh, I would call them abiotic conditions, in the sense that these are very high temperatures, very high temperatures, extremes of pH, and it's in the solid state. There's no water around. In the presence of TP, these conditions are more mild and more biotic, and overall information energy processing enables this emergence of these molecular strings across this diversity of environments, including mild ones where our life thrives. So it's in cooperation between the energy and information processing that allows us to see robust sorts of behaviors. So that's all I have, and I'll look forward to hearing from Carol. Thank you.
After Carol stop, we will have a conversation and Q&A. So Carol, please take it away.
So thank you again for inviting me. Oops. Is the microphone on? Okay, good. Thank you again for inviting me. It's a real pleasure to be here, and I had a wonderful time today talking to everybody. So I'm really grateful. So I'm going to take a very, shall we say, skeptical look at how much we know about life. Let me. Oh. Okay. Yeah. Okay. You want to get. I was going to say, that doesn't look like my slide. Oh, there I am. Okay. So I'm going to give you a skeptical account of the question, how do you answer the question, what is life? And I'm also going to be positive and tell you how I think you should approach answering the question, what is life? So there are two popular strategies for answering the question, what is life? One is, and you witnessed that John's talk earlier, is to define life. That's the most popular. There's an official NASA chemical Darwinian definition. Ironically, I'm going to talk very briefly at the end about the Viking lander experiments in the mid-70s, which were based on a chemical metabolic definition of life. So the definition of life is a moving target. And I don't think that's an accident. The other way in which people who don't like definitions try to answer the question, what is life, by appealing to a preliminary, truly general universal theory of life. So they usually supply, well, this is an essential part of life, and any theory of life would have to have this particular principle or this particular structure, et cetera, et cetera. What I want to argue is that basically the bad news is that neither of these is up to the task that's been assigned to it. And that gives rise to the question, well, what the heck do we do? And I have an answer to that. So let me tell you about the problems with defining life. So there is this very central, what I'll call a logico-philosophical problem with defining life, and that is that definitions are concerned with language and concepts. They really can't go beyond our current understanding and tell us about things in a mind-independent world of nature. So I like this joke saying there has to be more to life than occupying itself with its meaning. So it's a kind of a funny joke. So let me spell that out. There are, the kind of definitions that you're usually exposed to in a beginning logic class are things like bachelor. Bachelor means unmarried human male. But if you really think about this, this definition doesn't tell you about bachelors per se. What it does, it tells you what the word bachelor means in the English language. So it's really about concepts. And what it does is it proposes to give you a necessary and sufficient condition for the application of a particular piece of language. What do I mean by necessary and sufficient condition? Well, a necessary condition is a condition in the absence of which the word wouldn't apply, and a sufficient condition is a condition in the absence in which the in the presence of which the word would have to apply. So when you define bachelor as unmarried human male, what you're talking about is anything, if this is a proper definition, that's unmarried human male is a bachelor, and anything that's a bachelor is an unmarried human male. And yes, you can worry about the Pope and infants, and this is a product of the fact that language is vague. That is to say, people don't realize that, but language has borderline cases. There really are no truly proper definitions, but we try to get as close to necessary and sufficient conditions we can get. And the best job of getting close to that are what I call ideal, see, I'm trying to figure out which button here. Ideal, I call them ideal definitions because that's where we aim. But things completely change as soon as you try to define, let's see. So yeah, so ideal definitions work fairly well for answering questions about artificial human categories like bachelor, garbage, or chair. Why? Because, you know, those terms basically depend upon human conventions and concepts. And again, our bachelor, what better answer to the question, what is a bachelor, unmarried human male? You can't really do better than that. Yeah, the Pope's a worry, and you have to worry about human infants, and we can stick in adult, we can try to make it better. But the important point here is that that's a good answer. But once we get to questions about what people like to call natural kind terms like water, the situation changes completely. That is to say, ideal definitions don't work well for answering questions like what is water, what is temperature, what is lightning? And you may right away see why they don't, because the answer to the question, what is water, temperature, or lightning, that answer was discovered. It wasn't found by dissecting our concept of water, temperature, or lightning. And in fact, the concepts we've had in the past of some of these things have changed over time as our scientific theories have changed. So for example, consider what is water? People like to say, well, the answer to the question, what is water, is H2O. Now, I want you to imagine, as a thought experiment, that you are in the 17th century and you're a scientist. This is before the periodic table, before you knew about hydrogen and oxygen. And you're a 17th century scientist and you're attempting to answer the question, what is water, like we answered the question, what is a bachelor? That is to say, by defining it. And here's the difficulty. Your knowledge of water is limited to sensible properties of water, being wet, transparent, tasteless, and a good solvent. You don't know anything about hydrogen or oxygen or the periodic table. So when you analyze your concept of water, you're going to have to pick between being wet, transparent, tasteless, or a good solvent. It turns out that most of the substance identified by your contemporary scientists in the 17th century lack these properties. Consider muddy water. It's not transparent. Consider brackish water. It's not odorless. So which of the properties should you use to define water? Well, it turns out that the alchemists, I guess I'll get to Leonardo in a minute. Turns out the alchemists chose being a good solvent because they thought, gee, this is a really remarkable property of water. And so they identified solutions of hydrochloric acid and nitric acid as forms of water. They called them agua fortis and agua regia. And that was because they were even better solvents. But we now know that's wrong because they have a different chemical composition. But I think the best way to see this is this wonderful quote from Leonardo da Vinci. And he had a lifelong interest in water. Among his many other things, he was dabbled in science. And I just love this quote. And it expresses the complete perplexity that Leonardo has about the nature of water, which he's really wondering about. Because it's sometimes acid, sometimes strong, sometimes bitter, sometimes sweet, sometimes thick, thin. Look at this list. He has no idea which of these things to pick. It becomes noisome, laxative, astringent, sulfurous, salty. And of course, now we'd be able to explain that by saying there are all kinds of stuff in the stuff he's calling water. But of course, he doesn't have any concept at this stage of what constitutes contaminated water and what constitutes pure water. Why? Because we all call these things water and they're trying to look for something in common. And now I got ahead of myself before, sorry about that. So the alchemists, they chose solvency as the essential property of water and classified nitric acid and mixtures of hydrochloric acid, I already mentioned that, which are even better solvents than water as water. And we now know they were wrong. These chemical substances are different. They have different molecular compounds or different molecular ones. Water is H2O, for example, and nitric acid is HNO3. But nobody could have known this until the middle of the, in the latter part of the 19th century when we had the periodic table and people were doing chemical experiments and discovered up until the beginning, actually up until the end of the 18th century, with the work of Lavoisier, people still were bound by the old Aristotelian concept that water is an element. It's an indivisible element. And Lavoisier has this really long title to a paper where he actually said, no, it's not an element, it's a compound. He was able to split it into parts. So what we see here is basically that it's not like Bachelor. No amount of analysis of the perceivable properties of water familiar to a 17th century chemist could reveal that water is H2O. And the same is true of temperature, and the same is true of, these other so-called natural kind terms, but it's not true of Bachelor or garbage. Their meaning is only the meaning we give to them because they're artificial or conventional terms. So definitions don't work. That's the problem. You're not going to be able to come up with a definition of life because all you're going to do if you try to come up with a definition of life is now is you're going to just be analyzing our current concept of life. And I'm going to argue in a minute, our current concept of life is based on a single example of life. So what about a tentative theory of life? Well, this is what I'm going to say now cuts both ways. It cuts against definitions and tentative theories. The problem is an adequately general theory of life can't be based solely on life as we know it on Earth. Why? Well, I'm going to give you two reasons. First of all, you might think the amazing morphological diversity of life on Earth suggests that all the possibilities are represented here on Earth. I mean, look at it. Bacteria, dare I say viruses? Some people don't think they're alive. I happen to think they are, but I'll say viruses. Jellyfish, elephants, redwood trees, birds, you go on and on. This is enormous morphological diversity. And you might think, well, we have a very good general representation of life here on Earth. But that's not the case when you look at the molecular level. So the morphological diversity of life on Earth is basically very deceiving. Why? Because all known life on Earth, from bacteria to you to redwood trees, shares a microbial last universal common ancestor, affectionately known as LUCA, and hence represents a single example of life. And you might say, well, how do we get all this? How do we know? And that's covered up, LUCA just got buried. But how do we know that all life descends from this common ancestor? Well, I'm going to be developing that story, but the best way to say it is at the molecular biological level, all life on Earth's biomolecules are extraordinarily similar. So the proteins of all known organisms on Earth share the same 20 amino acids. The nucleic acids are remarkably similar of all life on Earth. They use the same genetic code. I'm going to develop in this a minute, the same coding strategy, the same nucleobases. You could go on and on. It's shockingly similar. All life on Earth is shockingly similar at the molecular level. And what you do is you basically analyze the differences and you find out that all life on Earth, if you project them backwards, it is descended from a common ancestor. And we can talk about that more in the Q&A. And I want to now talk about the astonishing similarities of all known life on Earth and show you that, talk about briefly, I only have 20 minutes, so it's really hard to do this, how it could differ at least modestly from familiar life at the molecular level. So, and we don't know how different it is. So let's talk about these molecular similarities. So proteins supply the bulk of the structural and catalytic material of all life, whether it's a bacterium, a redwood tree, or you. And here's just a picture of a protein. And what you see here are, they're just kind of polymers or chains of amino acids. And it turns out that all known life from bacterium to you uses the same 20 amino acids, but there are over 100 found in nature. Many of them are delivered by meteorites. Meteorites have delivered up to 70 amino acids. 8 of them are from these 20. The rest aren't. That's really interesting, actually. You can make them in hydrothermal vents. So there are many different, and atmospheric aerosols can be used to produce amino acids. So we have a lot of amino acids that could be used to produce proteins. And in fact, protein chemists have shown that you can make proteins, polymers, out of these non-standard amino acids, and they would be functional in the right organismal environments, but not exist on our planet. 100 are found in nature, and biochemists have synthesized non-standard proteins from abiotic amino acids that would be functional in the right organismal environments, which don't exist on Earth. Nucleic acids. So here's a picture of DNA. Nucleic acids store DNA and manage RNA, the hereditary information of familiar life. All known life uses the same nucleobases. Here they are. So there are 4 of them. This is actually not DNA. Yeah, that is DNA. So there's thymine, but these are the 4 amino acids in DNA. You have substitution of uracil for thymine and RNA, which is what does the translation process. We'll talk that in a minute. But all known life uses just these 4 in their DNA and basically substitutes in their RNA, which does the translation of DNA into proteins, uracil for thymine. And it uses the same, see these long chains, this is a sugar phosphate backbone of DNA, and RNA also has sugar phosphate backbone. They all use the same molecular structures. And we know that there are alternative possibilities for these structures. There are molecular biologists have come up with different, and we'll talk about that in a minute, different nucleobases. They've experimented with different phosphate sugar, mostly sugar backbones, but some people have thought they could fool with the phosphate backbones. We have PNA, we have various alternative strategies for storing material information, which might be functional in the right environments. All natural life uses the same triplet genetic code and the same coding scheme. So here's the coding scheme. You'll notice that I said triplet. So here are the bases. There, sorry, why did you do that? Microphone. What do we do with that thing that's in the middle of the, anybody know why that's there? Can I make that disappear? I'll just talk over it. Your default micro has changed to, how strange. But anyhow, the important point here is you can see that here's the triplet code, and it codes for various amino acids. And so you have here the 1-letter symbols. These are all the amino acids, and here is the coding scheme. So the coding scheme is redundant in the sense that you have no codes for more than 1 amino acid, but there are alternative codes that will code. So 1 amino acid can have different codes. So there's a kind of redundancy built into the system. That's exploited by synthetic biologists who are trying to create alternative. They try to stick in some abiotic amino acids where you have additional codes coding for the same amino acids. They knock out one and attempt to put in another. Yet, biochemists have constructed non-standard nucleic acids from modestly different nucleobases using alternative genetic codes. And here's just a picture of how it happens. You can see that where it says new, you can see where you've stuck in basically for a code, you've stuck in a new amino acid, and here is a chain of amino acids where now you're coding for these new amino acids. And I have a picture of Steve Binner because I'm on a grant with Steve and he's done a lot of work on this. And it was quite amusing because I told him I was going to give a talk at Wisconsin. Could he send me a nice slide? I wanted one like this. And he got all excited and invented this really complex slide that I couldn't possibly explain to you in this short a time and sent it to me. So I figured I didn't want to disappoint him if he asked me, so I added his picture. And I found this picture in a review. But it was really funny because he said, I did this just for you. And I'm like, Steve, it would take me 10 minutes, 15 minutes to explain this. So it was quite amusing. But then what's the moral of the story here? The moral of the story is that we know that all life on Earth is remarkably similar at the molecular level and its biomolecules. We also know there are alternative possibilities, that it could have been at least modestly different. And we don't know how different it could be. So this leads us to the zebra problem, or the N=1 problem in biology, which some people say is a trivial problem. I think is a really major problem. The N=1 problem in biology basically says you can't generalize to all of life, wherever it may be found in the universe, from a single example that we have reason to believe could be unrepresentative of life. So this is really important. It's not just that we have a single example, but we have reasons at the biochemical level to believe it could be unrepresentative, that life elsewhere might be fundamentally different in its biomolecules. And I don't have time to go into it now, but I talk about it in my book. It's also the case that, and this I think came out in the earlier talk, that people really, when they're talking about life, they focus on metabolism and reproduction. That goes way back to Aristotle. Aristotle had this big debate about whether life was best defined, and he wanted to define it in terms of self-nutrition or self-reproduction. And you still see this emphasis, which was made, I think, in the earlier talk about, is it, genetic-based reproduction that's essential to life, or is it metabolism that's essential to life? And that's a very multicellular eukaryote, plant and animal orientation, because if you look at the molecular level, the level of microbes, these things aren't quite so straightforward. And viruses are my favorite example because they can't self-metabolize and they don't even self-reproduce. They have to commandeer another cell's as a host in order to reproduce. And on these grounds, many biologists would reject them as not living. Some biologists who were really focused on Darwinian evolution will say, well, they evolved, so we should count them as life. But it's really important to see that that view, which is very, I think, based on multicellular eukaryotes or things like us, it's really much based on that. When you look at the micro level, things aren't quite as nice and clean as they are at the level of complex multicellular creatures. And the zebra analogy works as follows. Supposing an alien came down and they wanted to have a theory of mammals, and all they had to look at were zebras. What do you think they generalized? Would they generalize on its mammary glands? Only half of zebras have mammary glands. Or its stripes? And which is more important to being a mammal, the stripes or the mammary glands? Well, if you're just looking at zebras, if that's your only example of life, you're not going to think about mammary glands. You need more examples. And so I think that zebras provide us with kind of a good metaphor for the situation that I think we find ourselves in, because just because all life has a certain characteristic, all known life, maybe all Earth life is like the zebras, right? And the characteristics we think are true of Earth life, that are general to all of Earth life, are actually, shall we say, remnants from conditions at the time of the origin of life. And they aren't true in other worlds, on other environments. So that brings us to a dilemma. In order to answer the question, what is life, we need additional examples of life. It's a zebra problem. But how can we recognize truly novel forms of life, examples of life as we don't know it, if we don't have a definition or universal theory of life? Not at all obvious that we'll recognize it if we see it. We'll only recognize it if it's like sufficiently like our form of life. So they just hit it. I said, especially if it's microbial. You can't see that, but that's what it says. And most life in the universe, astrobiologists believe, are microbial. And so if you're going out and you're looking at Mars or even Titan, that's where I want to go. But if you're looking at Mars, you're going to be looking for microbes, because basically microbial life dominated Earth from 3.9 to 4.1 billion years ago. Shortly after, they think it emerged, pretty shortly after, geologically speaking, the Earth formed. And it stayed microbial until we first have the evidence of multicellular organisms, creatures with more than one cell, less than 1 billion years ago. So the Earth has been the haven of microbial life for much longer than it has been a haven for complex multicellular eukaryotes. So most astrobiologists say, well, we want to go and we want to look for microbial life because it's probably the most common form of life in the universe. And some biologists say we'll recognize it if we see it, but that's not at all obvious, especially if it's very different than our form of life. So here's the problem. If you search for life with a definition or tentative theory based on familiar Earth life, you run the risk of being blinded to truly novel life forms. We'll recognize alien life forms only if they resemble familiar life in the designated way. It's like looking for your keys under the street light. You lost your keys. So where do you go to look for your keys? Under the street light? Obviously, you could have lost them in the dark, but how do you look for it in the dark? It's dark. So you need a way to actually look for alternative forms of life without using really Earth-centric modes and tools of looking for life. So here's my solution. Use tentative versus defining criteria to search for anomalies instead of searching for familiar life via an Earth-centric definition or theory of life. So basically, what's an anomaly? It's a physical system that both resembles and fails to resemble life as we know it in provocative and unanticipated ways. What do I mean by that? Well, it's like the zebra case. So you're looking for mammals and all you've got is zebras. So of course you're going to look for stripes, but you're going to look for other things that aren't necessarily universal to all the kinds of things that you're looking for. So you might, if you're going to Mars and you're looking in a Martian environment, you might want to look for, the kinds of organisms that have adaptive strategies to desert environments that are extreme in the way Mars is. You're going to do something very different if you go to Titan or Venus to look for life. And so my thought is that what you do is you look for, you use tentative criteria, that is to say criteria that are based on Earth life, but they're not treated as defining. They're not treated as necessary and sufficient conditions so that either it fits them or it doesn't fit them. What you're doing is not looking for life per se. You're looking for something that looks biological in provocative ways, but also doesn't resemble our form of life in other particular ways, and also doesn't resemble in important ways non-living types of chemical systems. And you see this contrasts with the definitional approach, which identifies cases that don't conform to the favored definition as non-living, period. Once identified, an anomalous system becomes a focus for further scientific investigation. This contrasts with the definitional approach, which identifies cases that don't conform, because remember, we have necessary and sufficient conditions, to the favored definition as non-living, period. So if you're going with necessary and sufficient conditions, it's like the bachelor case, if it's 40 years old and married, it's not a bachelor, right? Because it doesn't fit being unmarried human and male. If it's a unmarried non-human, it doesn't fit. So with the definitions, if you really do have an ideal definition, it's either in or out. And when I'm looking for those cases that are basically provocative because they don't fit into either the non-living or the living category neatly. And those, I think, should become a focus for scientific investigation. And now, I don't think I have time, but I just wanted to quickly say, I think we have found anomalies. And I think NASA has found them. And if I had time, I would tell you about the mysterious Martian oxidant discovered in the Viking missions to Mars, because it has all the marks of an anomaly. And I think it should have been investigated more seriously. So thank you.
Just a reminder to our audience and our presenters that we do have about 100 guests joining us virtually. Please make sure that if you are asking a question or answering that we use a microphone.
Great, fantastic, thank you. So I'm gonna get us started and see if we can get the two of you in a bit of a debate.
Yeah.
I think
Oh, that wasn't part of the deal, was it?
Oh, yeah, we're trying. So John, I'll turn to you. And in your presentation, I'm wondering, do you think there are some specific kinds of chemistries that are essential for life? And if so, would you disagree with Carol about the dangers of trying to define life?
I guess I'm more, so the specific features that might be accessible through chemistry in the lab, which might resemble life and be quite different from what we think of as conventional chemistries, might be ones where the chemistry facilitates itself. So I'm interested in finding molecules that accelerate the reactions that give rise to their synthesis. Those are called autocatalytic reactions. An example of an autocatalytic reaction is if you take a cigarette and you light the end, then that will propagate. It will use the fuel of the cigarette to propagate spatially. And that's a reaction that uses the fuel to make more fuel, to make more heat that will continue to propagate. But once you've used the fuel, it's done. So if you want to keep that burning process going, you've got to continue to provide more fuel. We've got to keep on eating if we want to keep on being able to talk about life. So I think there are features that we're very interested in trying to recapitulate in the lab that entail chemistries that are not encoded in the specific building blocks, but we hope that we can find environments that enable these products to come out. Do I agree or disagree with Carol? on the aspects of trying to define life. I guess I don't spend a lot of my time trying to define life. I spend my time mainly thinking about, what do I spend my time thinking about? Trying to figure out what I'm thinking about. I think I spend most of my time trying to think about what are the ways that we can enable these sorts of emergent chemistries to occur. And I look to examples from virology, where I study the viruses, and I look at a variety of chemical and sorts of biological systems. I think in Carol's case, I would point at there are certain kinds of chemical reactions that are oscillatory. So you mix the chemicals together, and then they'll give you a light color, and then a dark color, and a light color, and a dark color. And it'll go back and forth, and you'll look at it and say, I've never seen anything like that. I wonder if it could be alive. And then eventually it just dies out and doesn't oscillate anymore. And these sorts of chemical reactions have the characteristic that I'm trying to enable my systems to do, but they're not continually fed. If you keep on feeding them, then they will continue to oscillate. And those are features that I think are the beginnings of something lifelike. That's the self-sustaining part, but there's not the adaptation that the NASA definition brought in, where conceivably information could be encoded, mistakes could be made, and the system could find new ways of doing what it does.
Great, thank you. So Carol, do you think that for those unlike Jonathan who actually do spend time trying to define life, do you think that is impeding our search for life?
So say
Do you think that if we spend too much time trying to define life, we are impeding our
Yes,
search for life?
yes, I do. Because what happens when you provide a definition, and scientists, people say to me, sometimes philosophers say, oh, the scientists don't really mean necessary and sufficient conditions. That's not true. If you listen to them, when they give their definitions of life, they have in mind necessary and sufficient conditions. And NASA wants a thumbs up or thumbs down. That's why the Viking landers are so interesting, because that's what they were looking for. They were looking for, and they didn't get it. They got something I'll call an anomaly. So I think the problem is that the Viking missions provide you with a good example of this, because what did they do? Well, they went with a chemical metabolic definition. It was agreed on by the PIs. And the PIs were really upset because they decided it was too weird a phenomena to actually say it was life. And they said, you're cheating, because we went with this metabolic definition and we satisfied it. But they said, but all these other effects, I wish I could have gone into them, we can't explain. So it must be an inorganic oxidant. And that's the thumbs up or thumbs down problem with definitions. They blind you to system. So if you have a system that's really different than life as we know it, particularly microbial systems is what you're looking for, you're not going to recognize it for what it represents if you go with a too Earth-centric a definition of life. So that's why I think you should be looking for potentially biological anomalies for the purpose, not of saying it's life or non-life, for the purpose of further investigation.
So if you were in John's lab, how would you advise John and his students and his collaborators and what they're doing?
I would advise them to not start their slides with a definition of life, which is what almost always happens in these talks, because scientists really like definitions, and they do know they're necessary and sufficient conditions. I've been in many a talk, and I think it would be much better to say, well, this system has these interesting lifelike properties, but these are the ways in which it deviates from some of our conceptions of life. Now, are these deviations serious? Or are these deviations that might lead us towards maybe this is an unfamiliar form of life?
In my defense, I would say that I put up the definition as a means to start the conversation. We're all on the same page
I
in
don't
some
mean
way.
to,
Right.
but almost all these talks do start with the definition.
Sure.
And it's rather intriguing. I think even starting with talking about definitions sort of skews your interest.
Okay.
In ways that are, I mean, as I said, if you look at I'm sorry I didn't be able to develop it, but the Viking missions, the definition was the key to shutting down the investigation of a mysterious oxidant, and life is an oxidant. But their view was, well, it didn't fit our metabolic definition of life, therefore it can't have been life. And so they came up with these various, maybe it was exotic states of iron, maybe it was hydrogen peroxide. The latest thing is perchlorate. None of them works very well. The perchlorate has problems. I don't have time to go into it. And they just persist in looking for inorganic oxidants. And so my view would be, if I had been advising the NASA team, gee, this is a potentially biological anomaly. We don't know whether it's biological. It could be abiotic. Let's go back and let's find out what it was. And they've never sent a mission. I once asked a NASA scientist why they hadn't gone and looked for it. And you know what he said? He was a high up NASA guy. He said, well, we didn't find life and that's what we were looking for.
So I'll ask one more question, then we'll open it up to the general audience. And John, so I'm wondering, do you regard any of the compounds that you synthesize as being alive?
Not by any means.
Okay.
So the
Okay.
question, are any of the compounds that we're synthesizing life? No, and I don't think we will create life in the lab, but I think it is possible that we will create precursors to what could be defined as life, because these characteristics are well, I don't think yet we have chemical systems that people have created in the laboratory that are not only able to sustain themselves when fed properly and also adapt. And I think there is that possibility, but I don't think we'll be able to call that life. We might call it life-like using conventional definitions of life. And I think it's very plausible that something like that will happen in our lifetimes, let's say next 10 or 15 years, because the tools of the chemistry for diversifying, also taking microbial life and altering it. So there's a top-down approach that Benner and others are using, or there's a bottom-up approach of mixing together components. So it's relatively easy to create complex soups. It's harder to analyze what you've created. So the analytical chemistry has come a long way in recent years. And now increasingly the computational. We and others are applying tools of machine learning also to try to figure out, can we predict, can we better understand the underlying mechanisms of these complex emerging chemistries?
Great, fantastic. Actually, I'm going to ask one more question. And if you've got questions, I think we're going to have you folks line up at the microphone here. So I actually ask this of both of you. And what can philosophers contribute to our understanding of life's origins?
So you're asking me?
Both of you.
Oh, both of you. Okay, so what can philosophy? So let me say that the way I view philosophy is that we can sort of be outsiders looking in, and I think there's a real advantage. We don't have all the dogmas that scientists have, that they kind of. A lot of these dogmas are, I could give you lots of examples, but what happens is they make you resist recognizing when something is anomalous. And you have a system, you're seeing something, it shouldn't be there. It's very easy to say, well, that's weird, but I'm sure it can be explained, and go on to something that's more straightforward. Tom Check's discovery of ribozymes is a really good example of that. So I think as a philosopher of science, what you can do is you can look at the work and point to things that are missed, such as problems with definitions, and the fact that definitions are thought to help you, but they actually can hinder your investigation of a phenomena like life. So,
John?
I agree with Carol that the philosophers bring in a very different perspective. And for me, the anomalies is something that if you're a scientist in the lab and you do an experiment and you have a hypothesis of how it'll come out and it doesn't come out that way, that might be an anomaly. And usually the good scientists will jump on that and say, this is kind of weird. We should do some more experiments to try to understand that. If you're not as deep thinking, then you might say, oh, that must have been just some dust from the room. Let's go and do something else, or let's try repeating it. So I think the anomalies are an important component. The difference from what Carol said was she would advise NASA to go back and look at this oxidant. I would advise NASA, hey, we know something about this. Let's study the anomaly in labs here on Earth if we possibly can. That would be a lot less costly, at least, and one could conceivably explore the chemistries of this particular oxidant. So I would take a different approach. A philosopher might take, say, we really got to go back to that particular site and look. But I think the philosophers absolutely offer a different perspective, and it's one that broadens. I have a BA in the liberal arts where we read philosophy. We questioned our perceptions. So I think it's important for us to be always. I think there's a tendency in sciences also to be either dogmatic or very fashionable. There are fashions, and one thinks about science as being highly rational and impartial, but we're also humans.
So can I just say one thing in response to that? So the problem, and they did do that, they actually tried to create in the laboratory conditions to show that particular oxidations, exotic oxidation states of iron could produce that. And they found that, yeah, exotic oxidation states of iron can do oxidizing, right? And the difficulty was that we weren't in situ in Mars, and we don't know that much about the complex Martian environment. And so that's the difficulty, I think, of running laboratory models at this stage. We just don't know all the conditions that are relevant.
Let me open up to the audience.
And if I could, while people are thinking about being brave enough to go to the mic, which you should just do, let me offer some questions that have come in
Please.
online, if I could. So we have two. We have right now one from Mark. Please comment on the definition of life as an information system which acts on resources and opportunities.
You want me to start?
Sure.
So I think, it's very popular among some circles to think of life, particularly artificial life people, to think of life as just this abstract algorithmic thing. I think that's what they're referring to. And to forget about the stuff that life is made of. So life on this view, if you can simulate Darwinian evolution on a computer, some of these people will claim what they have is a living thing inside a computer. The difficulty with that is that when you get to the stage where you don't know much about something, it's really tempting to go to the most abstract level and try to think of it in those terms. But as you become more sophisticated, you begin to realize that, chemistry matters, mass matters. I mean, if you look at, for example, some of the early work on water, saying it's a good solvent is pretty abstract, right? But saying it's made of 2 parts hydrogen and oxygen, now you're getting, in the nitty gritty. So my view is that the more abstract you have an account at the information theoretic level, the more likely it is that you don't know much about it if it's a natural phenomenon. Because I think if we look at the history, most natural phenomena, the stuff matters. You can't make water in a computer.
So this area of artificial life was picked up by computer scientists who tried to simulate processes of life. And they would have random number generators, they would have information processing. Many of the rules that we think about as associated with living materials of processes in cells, they tried to simulate with strings in the computer. I think I would agree that it's not in the substrate of the chemistries of life or materials that we would think of as life or lifelike. But I think there's value in such simulations in the sense that they can reveal principles that we might not have anticipated. In particular, in the artificial life, the random number generators could give rise to random strings that could act parasitically. That is, they could invade the artificial life system. And these are very virus-like, right? Without the artificial life system running, these random parasitic strings wouldn't be able to survive. So that's a principle that we see in living systems where these random parasitic strings like viruses arise. It turns out in my own research, we studied growth of viruses. And when viruses make progeny, they also make defective viruses that can invade normal virus infections. So they package like normal viruses. They replicate, well, they can't replicate in a cell because they're missing genes. But if they can replicate when a normal virus is growing, they can steal resources from the normal virus. So it might go on down that there are viruses of these viruses of these viruses. So I think there are principles that can be captured by such very artificial systems because they play with the rules and the implications of the rules, and they allow us to expand the way we might think about life.
So can I just have one
Yeah.
quick response to that? So there was a talk, I went to an artificial life conference once, and there was this chemist there. and chemist made the comment, he gave a keynote talk, I can't remember what his name was, but he said, he was so impressed by all of the simulations, he said, I just wish I could make the molecules do it. And I thought that was really a really significant response.
Sir, please.
Thank you. First of all, I want to thank the two of you for your great presentation. And I have two questions in mind. I'll direct my question directly to Carol directly. First of all, the one question that I had in mind related to planet Mars. You say that we should not be looking for life where we should be looking for other alternatives on how we can define life. That's what if I understood your question, because we cannot see life and use that as a way. We had to find other ways of life to define it. And you were mentioning a little bit about the anomaly that we think about in other areas. My question to you is, we're thinking about going to Mars in about 20 years, 30 years from now. So what should we be looking for if we're not looking for life as we know it on Earth?
So two things. First of all, I think we should just forget about definitions because if you look at basically physical theories like chemistry, we know a lot about water, but it doesn't define water. When you say water is H2O, that's not a definition. It doesn't supply necessary and sufficient conditions for water because a single molecule doesn't have the properties of water. If you get the purest water sample possible together, it hydrolyzes into ions and it's not pure H2O. And it's the polar nature of the molecule that allows it to have all these amazing properties of water, such as expanding when frozen, remaining a liquid over a wide range of temperatures, which is really important to life, and be a good solvent, among other things. And so, with those properties, if you had not been able to show that H2O was affiliated with those properties, you never would have said water was H2O. So basically, when you get a theory of, material substance, a chemical substance, you don't get a definition. What you do is you get a claim that this property here is a unique characteristic given the mechanism of the theory that distinguishes it from other chemical systems. So I think the whole search for definitions is mistaken.
Okay, so then I will relate that to my last question to you then. So the whole search of definition is a big problem. So my question to you is, what does life mean to you if I remove your hat as a philosopher?
What was the last part?
If I remove your hat as a philosopher, what does life mean to you?
What does life mean to me?
Yeah,
so I think that's a different question than the question, what is life? I'm a philosopher, so I'm really difficult. So by that, I mean, I think the meaning of life is a very different question than what is a biological thing called life. The work I've done as a philosopher of science has to do with, what is the thing that all living things from bacterium to redwood trees to me have in common. Now, you were asking me earlier about going to Mars or what to look for. What I want you to look for is potentially biological, that's important, anomalies for further investigation, not life per se. That would be my answer to that. But as for the meaning of life, I mean, consult a Buddhist. I mean, I don't mean to be flippant, but I think it's that different a question than the question about what is life. Yeah, your question's a good one. I mean, a lot of people think exactly along those lines.
Excellent. Yeah. Thank you very much.
Okay, thank you. Other questions?
We have a couple more online if people are still thinking about going up. And
Come
if
on
you
up
do
to
have
the
a
mic,
question, you need to go up to the mic, please. Another question came in for Carol. Is your view that there are no necessary and sufficient conditions for life, or only that we are presently unjustified in believing that there are such conditions?
Excellent question. And it's basically the question, do you really believe we can't define life? My answer to that is, I think they're probably necessary. I'm sure there are necessary conditions for life, but they're probably not simple-minded. Like being H2O is a necessary condition for water in some complex sense. It's not just being a single molecule of H2O. So I think, there's something that if life is what we call a natural kind, a natural category, then there's got to be something that bacteria, you, me, and all life in the universe, whatever and wherever it's found, have in common. But I think it's the zebra problem. We're going to focus on the strikes, and we should be looking at the mammary glands.
a follow-up that from another viewer online that might pull out what you just said is death as complete extinction of life, not the death of an individual, not a necessary and sufficient condition of life?
So I think people often think of, so there's a difference between death and non-life. And so I don't know that death is a necessary condition for life. It depends how you define biological individuality, and there's certainly certain bacteria and certain systems that it's not so clear that you want to say they die. So I think that's a complex multicellular organismal response. But I think we really have to distinguish life, non-life from death. And people often blur those two. Do you want to say something about that, John?
No, I think that's that.
Yes, please.
Yeah, thanks for being here. Really, very, very interesting. I think this is consistent with what you've been saying, but you can educate me if it's not. We have, it appears, a fairly well-defined definition of life, at least as it exists here on Earth. And outside of Earth is another question. And I think what you're saying is, why don't we just take the approach that says, don't worry about whether there's life outside of Earth or not outside of Earth. Let's just see what's out there, learn what's out there. And then once we learn what's out there and gather sufficient information, then our understanding of what life is can evolve based on what new information we gather outside of Earth. Is that a fair summary?
I would put it a little differently. I want us to look for potentially biological anomalies. There are lots of anomalies out there.
Mhm.
I mean, they've discovered that Pluto has, plate tectonics with glaciers, and they've recently discovered an exoplanet that has iron rain. We're going to find lots of weird stuff out there. And Mars, there's things that people can't explain. So I don't want to just look for any old weird thing. I think we should look for specifically those kinds of potentially biological anomalies in the sense that they have strikingly similar characteristics in some ways to life as we know it, but they differ from it in other ways. And we want to go up with a suite of criteria that we expect some will fail and some will succeed. And if enough of them are met, then we go, this system's very strange and we should take another look at it.
Some you may not understanding. So to that extent, you think there's going to be at least something in common.
Yeah.
Life
clearly-
outside the Earth is going to have at least some commonalities with life on Earth. Is that what you're
Yes,
saying?
but we just don't know what ones, it's
Oh,
like
I
the
see, I
zebras,
see,
right?
okay.
So
Okay.
what do zebras have in common with all mammals? Not their stripes, but if you've never seen a non-striped mammal,
Right.
you're gonna,
right.
and all the mammals you've seen are striped, then you're gonna think stripes, but it's mammary glands. And without other examples, you're never gonna hit on the mammary gland's importance.
So you're not open to the idea that life outside of Earth could be such that it doesn't have anything in common with life as exists on Earth?
Oh, it has to have something in common with life on Earth if life is a natural category.
Okay, if life what?
If it's like water, a natural category,
Okay,
it has to have something in common. My problem is I don't know what it is.
right.
And so I think we have to have our search strategy more open so that we can catch stuff. If you go with a definition, it's thumbs up or thumbs down.
But your search strategy is going to have to pick some elements of life on Earth to incorporate in order for the search to proceed forward, isn't
But
it?
it has to do it as a tentative, not defining criteria. And it has to have a large suite of criteria, some of which are universal to Earth life, but others of which are criteria that are specific to certain kinds of extraterrestrial environments you're searching, because at Earth, we've seen they're adaptive, but they're not common to all of life. So you have to have if you're going to Mars, your search criteria, your tentative criteria are going to be different than if you're going to, say, Titan. So.
Okay, and the only other last comment I make is this fascinating. Thank you for the information. Is that from a philosophical perspective, I think what you say is really, really enlightened. But from other perspectives, for instance, a political perspective, I mean, if you go to Congress and say, we
Oh,
want
I know,
$10 billion
I know.
because we
I
think there
know.
might be
I'm a
anomalies on
philosopher. I don't
Mars.
have
That's
to-
right.
I don't have to worry what they have
Right,
to worry about.
okay.
They have to worry about funding. I don't have to worry about funding.
Right, good. That's, yeah.
And I get a little money from scientists who want to hear what I have to say, but they don't want to talk too much in the halls of Congress about what I say.
Thank you very much.
Let's have we have another question online.
Sure, there are two. We don't have time to get to both of them. I'm sorry. I'm going to take the first one that came in. Can you comment on GMOs and other products of synthetic biology? Are those considered life? And then what do we do about the whole question around viruses?
I can try GMOs, genetically modified organisms. So you are taking what we would accept as living organisms and changing the genetic material in them. And oftentimes these are to create products that have properties that we deem useful. Are they life? I would say yes, because they are reproducing. They're capable of Darwinian evolution. They are derived from life. They're not natural life in the sense that humans played a role in their creation. So I would say they're modified or engineered life, or the field of synthetic biology is nevertheless working in the medium of biological organisms. Viruses, people ask me, okay, I've heard about this controversy, are viruses alive or not? And I usually say, well, if you think about the properties of living organisms that are able to adapt and evolve, yes, they are definitely able to adapt and evolve. We can build trees for how they've evolved. We can look at the coronavirus and alpha, beta, delta variants, and these properties of how they evolve are using the same sorts of machinery for how cells evolve. In that sense, they are alive, able to evolve. In the sense of taking energy from their environment, you can feed a cell a candy bar and it'll grow, it'll reproduce. You cannot feed a virus a candy bar. The candy bar is not living unless you've got bacteria growing on, and then it's not a then it's the living entity, it's the living cell, or the host that the virus needs in order, a much more complex organism. And you could also call a infected cell as even more complex than the virus or the host cell itself. So I think cell that is able to feed on simpler compounds, extract energy, and use the biosynthetic machine over the cell to replicate itself.
Great, thank you. And so please join me in thanking John Carroll. Wonderful presentation, great discussion. Thank you very much. And thank you all for coming out this evening. I hope you enjoyed it. The next one is in a month or so. On consciousness. So please come back. I think y'all have a wonderful evening.
Fun discussion. So thank you.
Thank you.
I'm going to have it.